Hi everyone, I am Dr. C.S. Kulkarni, MD, and I am going to teach medical biochemistry. Before I start the topic, please like, share, and subscribe the channel. In the last session, we had dealt with factors influencing enzyme activity. In today's session, we will continue with the ribozymes. In the first part of this chapter, it is stated that all enzymes are proteins. Invariably, all rules have exceptions. Ribozymes are RNA molecules with enzyme activity which catalyze the cutting of nascent mRNA or primary transcript. Coming to the next point, enzyme assays and units. Enzymes are assayed by taking the serum sample and adding suitable substrate. After the incubation period, the mixture is assayed for the product form. NAD plus or NADP plus dependent enzymes are assayed by spectrophotometry, as the reduced form is of these coenzymes will absorb light at a wavelength of 340 nanometer, but their oxidized form will not absorb the light. The reaction of our interest can be suitably coupled with another reaction utilizing NAD plus or NADP plus, and then the reaction rate can be assessed. Enzyme activity is expressed as micromole of substrate converted to product per minute under specific assay conditions. One standard unit or one international unit of enzymatic activity, that is U, is the amount of enzyme that will convert one micromole of substrate per minute per liter of sample. It is abbreviated as units per liter. The modern method of expression of enzyme activity is catals. One catal, that is catalytic activity, is defined as the number of mole of substrate transformed per second per liter of sample. When expressed in catals, the activity of different enzymes can be compared. Catalytic efficiency is expressed as the ratio of kcat upon km. The specific activity is the number of enzyme units per milligram of protein, that is Vmax divided by protein concentration. It is measure of the purity of the preparation. Turnover number is the number of substrate molecules transformed per unit time by a single enzyme molecule or by a single catalytic site, that is Vmax upon number of enzyme molecules. This is estimated as purified preparations of enzymes. For example, the turnover number of catalase is 40 million. For carbonic anhydrase, it is 4 lakh, whereas for humarase, it is 800. Coming to the next point, body metabolism in controlled enzymes. In a pipe, theoretically water can flow bidirectional, but practically the flow is unidirectional. In the same way, although most of the individual enzyme activities are readily reversible, the pathway as a whole tends to lead towards the final product. In the reaction series, A leads to B and B leads to C. As soon as C is produced, it is utilized for something else, so that the reaction as a whole tend to go in the forward direction only. True equilibrium of chemical reactions in the cell is achieved only when the cell is dead. In other words, the living cells avoid the state of chemical equilibrium. At the same time, if we analyze the particular metabolite, the concentration is more or less kept constant over long periods. This is called steady state system. For example, blood glucose level is kept within a narrow range because the factors which tend to increase the level are at a dynamic equilibrium with the factors which try to decrease the level. Deficiency of the enzyme or its activity to, due to genetic defects, that is, inborn errors of metabolism. Such deficiency may be due to genetic mutation or by infections. Coming to the next point, drug metabolism. Drugs act on enzymes present in human beings or in pathogens. Penicillin blocks cell wall synthesis in bacteria and by irreversibly binding of the enzyme transpeptidase. Many bacteria, however, produce beta-lactamases that hydrolyze the penicillin. Another example is that 5-fluorouracil is metabolized inside human body into deoxyfluorouridylic acid, which inhibits thymidylate synthase and hence acts as anti-cancer drug. Enzymes are widely used in food, pharmaceutical, and chemical industries. Bacterial enzymes from time immemorial accomplish fermentation of food items. Curd from milk is by bacteria Lactobacillus acidophilus. Yogurt or cheese by Streptococcus thermophilus. Fermenting rice and black gram by Lucanostoc acanthoides for preparing delicious dosas are good examples. In washing powders, enzymes are incorporated into stains from clothes. Coming to the next point, drug designing. With the help of computer programming, it is nowadays possible to get an idea of the three-dimensional structure of the active site of the enzyme which fits with the substrate. With this knowledge, research workers could make theoretical models of hundreds of different inhibitors. They continue to act on the particular substrate and do not dissociate between the repetitions of the catalytic activity. For example, DNA polymerase, RNA polymerase, glycogen synthase, fatty acid synthase, etc. Coming to the next point, multi-enzyme complexes. Enzymes are diffusion limited, meaning that the rate of reaction is limited by the rate at which the substrate molecule diffuses through solution and reach the active site of the enzyme. In a series of reactions, the first product of the enzyme is diffused into surrounding medium and later it reaches the second enzyme. This may act as a hindrance for the smooth muscle and efficient work of the enzymes may be in a metabolic pathway. But nature circumvents this problem by keeping all the enzymes of a reaction sequence into a multi-enzyme complex so that the first product of the first reaction is immediately transferred to the second enzyme and so on. Examples are fatty acid synthase, pyruvate dehydrogenase, alpha ketoglutarate dehydrogenase, acetyl CoA carboxylase, glycine cleavage system, pyrimidine nucleotide synthesis. Coming to the next part, sequential reactions or single displacement reactions. In such cases, both substrates are first combined with the enzyme to form a ternary complex, then catalysis is followed. Most of the NAD plus dependent reactions follow this principle. Coming to the next, ping pong reactions or double displacement reactions. They involve a transient modified form of the enzyme. The group undergoing transfer is first taken from one substrate, added to the enzyme, Subsequently, the group is taken from the modified enzyme and added to another substrate. This is, examples are aminotransferases. Coming to the next point, single molecule enzymology. Recent advancement in nanotechnology have made it possible to observe catalysis by individual enzyme and substrate molecules by fluorescence microscopy. Even the rate of single catalytic events and sometimes individual steps in catalysis is measured by this process. Coming to the next point, iso-enzymes. They are physically distinct from forms of the same enzymatic activity. Multiple molecular forms of an enzyme are described as isoenzymes or isozymes. Study of the isoenzymes is very useful to understand diseases of different organs. If the subunits are all the same, then the protein is homomultimer, represented by a single gene. If the subunits are different, then the protein is said to be a heteromultimer, produced by different genes. Coming to the next point, isoenzymes may be formed in different ways. They may be products of different genes, that is more than one locus, in case which in which case they are known as true isoenzymes. The genes may be located on different chromosomes. Example, salivary and pancreatic amylase. In certain cases, all the different forms are present in the same individual. Example, lactate dehydrogenase has 5 isoenzymes and all are seen in a person in the population. The same locus of the gene may have different alleles, that is, alternate forms. Such allelic isoenzymes are called allozymes. In this case, only one form will be present in one individual, but all the different forms will be seen in total population. For example, more than 400 distinct forms of glucose 6 phosphate dehydrogenase have been identified. All of them are produced by the same locus on the X chromosome. When isoenzymes are due to variations at the single locus occur with an appreciable frequency, that is more than 1% of the population, it is called as polymorphism. Molecular heterogeneity of enzymes may also be produced after the protein is synthesized, that is, post-translational modification. This is called as isoforms. Example, sialic acid contained in alkaline phosphate isoenzymes. Different types of isoforms may be seen in different individuals, in the same individual. Here ends the session. Kindly like, share and subscribe the channel. Thank you.